Hello everyone, how are we doing today? Welcome to today's video where we're going to be going over an example problem going over gene expression. So the last couple chapters we've gone through transcription, translation, to then go from DNA to a protein. So let's look at an example of going through that. It's not, this stuff isn't crazy too hard, but it's really easy to make mistakes and screw up a simple, base, and then that causes a mutation, and then you don't create the proper amino acid. So let's go through this little example problem here. Remember, always work through these example problems first on your own. So I'll read through the whole problem here, and we'll get started. So the following represents a segment of a piece of bacterial DNA. This is significant. Bacterial DNA containing a gene. The template strand is labeled right there. Assume that the first nucleotide shown is the transcription start site. So there's a little arrow pointing that direction, and that tells you the directionality. But if this is given to you as a template strand and you can see the directions, the 3' and 5' here, it also tells you the direction that it goes. The first part to this problem is give the messenger RNA sequence that would be produced from this gene by transcription and label its 5' and 3' ends. Then, using the codon table provided below, give the amino acid sequence that would be produced from the messenger RNA during translation. Also label the N and C terminal. So if you go down here, I provided the amino acid table for you as well. And then last part is, how would this sequence differ in eukaryotes? So we'll get to that one at the end, even though you can technically answer it now. All right, so first step here is give the messenger RNA sequence that would be produced. Label its 5' to 3' end. So I tell you this is the +1 site, or the start site right here at this T. So how do we start this? Well, if we're doing, anti-parallel and complementary base pairing, if this is 3', the start of the sequence is going to be 5'. And I also told you this is the template strand. So you're going to be forming base pairs with these bases here. And wherever you would have an A binding with a T, there are no T's in RNA. So that's a uracil. Now, a hint. You could go through and be like, okay, T with A. That makes sense. So the first one would be an A, G is a C, G, C. So you could do that. You could think of the base pairs as you go down through here, or you can look at the non-template strand right here. So the upper strand here is a non-template strand. Also might be called the coding strand because it directly codes for what the messenger RNA strand will be, except you just have to remember that T's are used. So you don't have to think about the base pairing and maybe accidentally make a mistake here. So just a little tip here where you just easily convert these. So just remember, wherever there's a T, you just think U right there. But so I'll write this all out quick. I won't go through each one. So I'll be right back. Alrighty, so here is the strand, and we labeled the 5' and 3' ends. Again, this is directly the same as this strand up here, and it forms complementary base pairing. Well, exactly the same except for the uracils, and it forms complementary base pairing with this strand here. So now we have our messenger RNA sequence, and since this is in bacteria, we don't have to worry about post-translational or post-transcriptional modifications, something like adding the poly A tail, the 5' cap, and splicing. So this is just the direct gene location. So now we have that. Next problem is, using the codon table provided below, give the amino acid sequence that would be produced from the messenger RNA during translation. Label the N and C terminals. Okay, so first step is, which one is first? So remember, for an amino acid, you have the center carbon, the alpha carbon, you have the carboxylic acid group, you have the amino group, and you have, there's a hydrogen here, and then here is the R group, or the whatever that would change for whatever the 20 amino acids are. So remember, the N, the amino site, is the N terminal, and the C is the C terminal. Now, when you form that chain of amino acids, where every circle here is a separate amino acid, the peptide bonds are adjoining them, one of these has its end, it's oriented a certain way. And the first amino acid is always N terminal. So here, the N is always first. So if we're listing a chain of amino acids, this would be the amino end, and this would be the carboxylic acid end down here. So N and C terminal, because when the peptide bond forms, it attacks this oxygen and then the neighboring amino group here. So a carboxylic acid will always be on the N, and the amino group will always be first. So that's the first part here. Now we got to start writing in what our amino acids are. So you want to look through. So down here on the table, the sequence you want to find first is the start codon, AUG. So we go up through and we see ACC, GUA. Oh, there it is, AUG. Right there's our start codon. Okay, great. So then you can kind of highlight, the order. So there's one codon, there's two codons, 3, 4, 5, 6, 7. Oh, you don't want to screw up your reading frame, so this should be UGA, 7, 8, 9. Okay, great. Now you want to figure out where you're going to stop. So one of these better be a stop codon. If you didn't get a stop codon, you wouldn't be able to, the ribosome wouldn't fall apart then. It would be stuck there. So if you have a mutation that affects the stop codon for a gene, it wouldn't be able to finish translation then. It would just get stuck in the ribosome without being able to be released because it needs those release factors present. So here are the 3 stop codons, UAA, UGA, and UAG. So then look up here and see if any of those matches. So UAA, UGA, and UAG. I see you got to look at which one's here. So here's UGA. UGA looks like our stop codon. So boom. Stop codon. Now, interestingly, there is another AUG that appeared down here. But again, if, say, that because of this 5' up here and the AUG being right there, there are some other consensus sequences that play a role here. So remember the Shine-Dalgarno sequence. So that still plays a role, and that AUG needs to be upstream from that Shine-Dalgarno in bacteria. In eukaryotes, remember, it's called the Kozak sequence. So this one still wouldn't be selected down here, but that's beyond the reach of this example problem. More info can be found in a lecture video for that one. All right, so there is our little, those are our bases then. We have 1, 2, 3, 4, 5, 6, pretty much 5 amino acids here. So now let's go through and figure it out. So AUG, of course, is the start codon for methionine. Now there's something unique. This is bacteria. So because this is bacteria, we know the first one is that N-formyl methionine. So you write this one as FMET. And you could just use the 3-letter abbreviations here. That's fine. So we can answer this next problem next. So how would the sequence differ in eukaryotes? FMET would be just a regular methionine. So there's no FMET in eukaryotes. So make sure you read the problem carefully, because if you don't write FMET for a bacteria, it's technically wrong. It's a slightly different form of methionine. So it's just a methionine in eukaryotes. Okay, so next one is UUU. That's a phenylalanine, or PHE. And you just go through and fill them all in. So phenylalanine, and I'll go through this just so we can practice together. AAA, how you find it. There's AAA, lysine. Next one is CAA. So CAA, that's glutamine, or glutamate, GLN. Next one, or glutamine, next one is, we just did CAA, ACA. ACA is ACA, threonine. And then you don't have to write the stop codon. So then UGA is a stop codon, so that wouldn't be an amino acid, so you wouldn't write anything there, and then you just have your C terminal. So right there's your answer. So that would be the final answer for that one. And again, so just the hardest part here, finding the start codon. You don't want to find the wrong start codon. So like if you look down here, there's another AUG. That would be a wrong reading frame then, because it'd be the last one there. But just this problem just doesn't show the, the Shine-Dalgarno sequence that comes before it that helps orient itself in the right position for the stop codon on the small subunit of the ribosome. So be careful. Make sure you find the first AUG and not one that's upstream. All right, so that's all I have for this example problem here. I know, not a crazy hard one, but we did talk about the, deeper things into the problem, like the, different sequences and, basics of the amino acid stuff here at the N terminal and C terminal. But yeah, the key thing is make sure you know the difference between F-met and met for the methionine and the difference between eukaryotes and prokaryotes, and read the problem carefully up here as well in regards to that. Alrighty, well, that's all I have for this video. If you have any questions, please let me know, but see you all next time. Bye-bye.